In this video, we're going to talk about the parasitic amoeba, or let me say, we're going to talk about the parasitic protozoan. And the representative of this protozoan is amoeba that belongs to the class Rhizopoda and the subphylum Sarcomastigophora. It belongs to Sarcodina, right? Now, let's talk about the general characteristic of the protozoan first before we move to the parasitic amoeba. Now, we have to understand that the terms protozoan are gotten from two words, that is protos, that is first, and zoon, that is animals, and it was given by Goldfuss. And according to the five kingdoms classification system, protozoan belongs to phylum Protozoan and kingdom Protista. Now, protozoan are small and they are generally microscopic. They are the simplest and the most primitive of all animals, having a very simple body organization. Hence, I can say they are in protoplasmic level of organization. So they are in what? They are in protoplasmic level of organization. They are all unicellular and they can be uninucleate or binucleate. Now, the body shape is variable. They can be oval, they can be spherical, they can be solitary, and they can be colonial. Now, the body protoplasm is differentiated by the outer ectoplasm and the inner endoplasm. So let me add a page. If you have something like this, let me take for an example. Let me take this as an example. This is an example of a typical protozoan, and an example of this is Paramecium caudatum. Even though it is not parasitic, but it's also a what? It's also a protozoan. This is what? This is the cilia in the body. Hence, we can say they have what? They have organelle of locomotion. They have nucleus. They can be uninucleate or what? Or binucleate. So let me take for an example. This one is binucleate. You understand? Paramecium is binucleate. And they have contractile vacuole. They use contractile vacuole for what? They use contractile vacuole for osmoregulation. Are you getting me now? They have contractile vacuole for osmoregulation. They have food vacuole for what? For storing what? For storing their food. Are you getting me now? Now, the most complex protozoan is Paramecium. You understand? It's the most complex, but it is not parasitic. It is free living. Hence, we can say protozoan can be free living, can be saprophytic, can be parasitic, and can be commensal. You understand? So they can exist in what? In different forms. Are you getting me now? Their body is covered with pellicle, most of them, except for the amoeba. Except for the amoeba, this thing. Their body is not covered with pellicle. But other parameciums, their body is what? Covered with pellicle. And this pellicle gives their body a defined shape. I get me that. It gives them a defined shape. Now, I said their body is differentiated into the outer ectoplasm, that is this, the outer ectoplasm, and the inner endoplasm, which is somewhere around here. Are you getting me now? So inside the inner endoplasm, the outer and the inner endoplasm give rise to what? To cytoplasm. So inside the endoplasm, we have the what? We have the organelles. You understand? Hence, I said they were what? They are in protoplasmic level of organization. Most of them, or let me say all of them, or most of them rather, they are aquatic, while few are what? Few are parasitic. And this is just like they have, like I said, they have organelle of locomotion, like pseudopod in amoeba. Yes, pseudopod in amoeba. We have cilia in paramecium. We have flagella in mastigophore. You understand? So let me take for an example. If you have something like trypanosoma. Trypanosoma looks like this. Don't mind my drawing. It has a kinetosome, you understand, and it has an undulating membrane that runs the flagella. Are you getting me now? So this is what? This is the flagella of it, and the what? The kinetosome is here. So the flagella arises from the what? From the kinetosome. You understand? If you look at it, it has a pellicular shape and it is spindle shaped. So they have what? Their body is covered with pellicle. You understand? So this is just an introduction to what? An introduction to phylum Protozoan. You understand? And we can say that they can be classified into different classes. You understand? We have the Sarcomastigophora, we have the Sporozoan, we have the Celiophora, we have the Nidospora, we have the Apicomplexan, and other stuff like that. But like I said, what we are talking about today is parasitic amoeba. What we are talking about today is parasitic amoeba. So let's go to the introduction. most species of amoeba, except the free living form, they are parasitic. You understand? And they inhabit the intestine of man and other vertebrates, causing a disease known as amoebiasis. So this is the disease that they caused. You understand? And the amoeba that causes disease are related to four genera: Entamoeba, Iodomoeba, Endolimnax, and Dientamoeba. You understand? And we have different examples of these animals. You understand? Under Entamoeba, under the genera of Entamoeba, we have the Entamoeba histolytica, Entamoeba coli, Entamoeba dispar, Entamoeba gingivalis, Entamoeba amani, Entamoeba moskowski, and Entamoeba puleki. You understand? And under Iodomoeba, we have Iodomoeba buski. Under Endolimnax, we have Endolimnax nana. And under the Entamoeba, we have D. fragilis. So these are representative of what? Of each genera. Are you getting me now? So we move from there. You see, Entamoeba histolytica and Entamoeba dispar, they are identical to each other, which means this histolytica and Entamoeba dispar, they are identical to each other. You understand? And they infect man and other mammalian hosts, like they can infect cats, they can infect dog, they can infect primates. They causes what we know as amoebic dysentery. That is what they cause, amoebic dysentery or amoebiasis. You understand? Now, this, like I said, what do parasites do? They cause harm to their host. But not all parasites causes disease to their host, because we have two types of parasites. We have the pathogenic parasites and the non-pathogenic parasites. The pathogenic parasites are those ones that causes disease. The non-pathogenic parasites are those ones that does not cause disease but they cause harm. But now, we now have, like I mentioned, I said we have four genera of amoeba that's associated to human amoebiasis. That is Entamoeba, Iodomoeba, Endolimnax, and Dientamoeba, right? Now, we now have, okay, we don't move to this one yet. We haven't moved to this one yet. Okay, let us take the first representative. That is Dientamoeba fragilis. Dientamoeba fragilis is an harmless commensal that inhabit the large intestine of what? Of a man, and they feed on debris, like what is inside the what. And one thing you have to note in this is that they are what? They are commensals. Hence, they do not cause disease. The only thing that they do is that they feed on nutrients, vital nutrients in humans, in their host, but they do not cause disease. That's why we call them commensals. They are not pathogenic parasites. Are you getting me now? Now, it is known that it is the only known species of this genus and affects 4% of humans. So, in the Entamoeba genus, we have only one species, and that is D. fragilis, and they exist as trophozoites alone. There is nothing like cyst. So they exist as trophozoites because initially an amoeba can exist in two forms. They can exist as trophozoites or a cyst. They can exist as what? Trophozoites or a what? A cyst. So we have two forms, the trophozoite form and a cystic form. You understand? So these are, but the Entamoeba fragilis can only exist as what? As a trophozoite. They don't exist as cyst, and they are delicate, and they disintegrate rapidly into in fishy. That is where they got their name, fragilis, fragile, something that is fragile. You understand? So they are delicate. So once they are passed out in the fishy like this, after a little while, they are going to what? They are going to disintegrate. So that is the word fragilis, fragile. You understand? Now, what are the features of the trophocyte? There is presence of two nuclei. I'm going to show an image that actually shows this. You understand? They have two nuclei. Endosome is eccentric, and they are about 6 to 12 micrometer in diameter. Now, what do we mean by endosome is eccentric? That is the question. So what we mean by endosome is eccentric is if we have let me say we have an amoeba like this, for example. Don't mind my sketch. So this is the center of the what? Of the cell, Abby. Is the center of the cell. So if they said something is eccentric, it means it is not at the center. So it is somewhere around here. You understand? The endosome is somewhere around there. Are you getting me now? So that is what we mean by it is eccentric, and they have food vacuoles. You understand? They have food vacuoles all around. So that is just it. So that is the what? That's the features of their trophocyte. Now, moving from there to Entamoeba coli. Entamoeba coli often coexist with Entamoeba histolytica. The both of them usually exist. So Entamoeba coli is a commensal, but Entamoeba histolytica is a parasite. Are you getting me now? These are the difference between the both of them. Entamoeba coli is a commensal. Entamoeba histolytica is a what? Is a parasite. And what we mean is that whenever you see Entamoeba histolytica, whenever you see Entamoeba coli in the body of someone, like in the fishes, in the fecal matter of someone, just know that there is opportunity or there is probability that we are going to find Entamoeba histolytica, because most, like two of them often, like they often coexist. That is Entamoeba histolytica and what? Entamoeba coli. That is E. coli and what? E. histolytica. Now, let's talk about the features of their trophocytes. They also have trophocyte form. They are about 20 to 30 micrometer in diameter, and they are identical to Entamoeba histolytica, but their nuclei is different. Now, the endosome of Entamoeba coli, the endosome is eccentric. That is, it is not at the center, but the endosome of Entamoeba histolytica is at the center. You understand? It is at the center. The chromatin lining the nuclear membrane is coarse with larger granule than that of Entamoeba histolytica. And the food vacuole of Entamoeba coli contains bacteria and um, intestinal symbionts. That is what it contains. It contains bacteria and intestinal symbionts. And Entamoeba histolytica consists of blood cells. So this shows that in Entamoeba coli, they only feed on food debris. You understand? While Entamoeba histolytica feed on what? Feed on blood cells. So their food vacuole consists of what? Consists of blood cells. I'm going to show an image that actually back that up. You understand? I'm going to show you images of Entamoeba histolytica and Entamoeba coli. Are you getting that? Now, look at Entamoeba histolytica. This is an image of Entamoeba histolytica. Can you see the wood vacuole? Look at the wood vacuole here. You can see something like reddish inside. So let me just try to zoom that in. Let me try to zoom it in. And look at this. Can you see something red inside? That is what? That is the ingested red blood cell. Like, look at this. Once they ingest the red blood cell, they what? They digest it. They break it down. Once they break it down, they store it inside the what? Inside the food vacuole. Are you getting me now? Can you see? So that is what? That is um that is Entamoeba histolytica. Can you see the cytoplasm? The cytoplasm consists of what? The food vacuole, the nucleus, the ingested red blood cell. They can also ingest bacteria as well. In some cases, they can ingest bacteria, and they have pseudopod. What do they use pseudopod for? To move. You understand? So that is the image that shows what Entamoeba histolytica. Are you getting that? Now let's talk about Entamoeba gingivalis. From the word gingiva, like gingivalis, they are found mostly in the gingival pocket near the base of the teeth. That is where they got their name, Entamoeba gingivalis. So it is an harmless commensal. It is also found in the teeth. It is found on the teeth surface, in the teeth gum, gingival pocket of the what? Gingival pocket near the base of the teeth, and the crypt of the tonsil. They can also find the crypt of the tonsil. Now they have no cyst. They can only like trophocytes. You understand? They have only trophocyte form. Hence, they can be transferred from one host to another through kissing, hmm, droplets, eating utensils, or sharing of toothbrush. That's how they can be like transferred. You understand? And it is estimated that 95% of people without hygienic mouths are actually prone to have this commensal in their teeth. You understand? Remember, it's a commensal. It does not cause disease. Are you getting me now? Now let's talk about their feature of their trophocytes. They have numerous food vacuole, and the food vacuole consists of what? Of cell debris, and in some cases it can consist of what? It can consist of um blood cells. You understand? It can consist of blood cells, and the nucleus is about 2 to 4 micrometer in diameter, and it is small. You understand? It is very, very small, and um they have a nearly central endosome. You understand? And one thing you have to notice, like one thing I forgot to mention there, is that the cell is about 10 to 20 micrometer in diameter. Their cell. I forgot to write it here. 10 to what? 10 to 20 micrometer in diameter. Now, going from there to the endolimnax nana. Endolimnax nana, and that is the last representative that we are choosing here. Now, the endolimnax nana is also an harmless commensal. It is an armless commensal that lives in the large intestine of a man, feeding on bacteria, and it has two forms. It has the cystic form and the trophozoite form. It has what? The cystic form and trophozoite form. Now, the features of the cyst is that it consists of a glycogen granules. It consists of what? Glycogen granules. And if you're talking about glycogen granules, it means it store food in form of glycogen. So they have food vacuole in trophozoite and they have glycogen vacuole in what? In cyst. You understand? So the trophozoites, they are often tiny, about 6 to 15 micrometer in diameter. Can you see the trophozoite? They're about 16 to 15 micrometer in diameter, and the nucleus is small and it's eccentrically located endosome. You understand? And the food vacuole consists of what? Are consisting of what? Of debris and bacteria. You understand? That is what they feed on. Remember, they are what? They are commensal, and they are found in what? In large intestine of a man. Now, look at it. This is an image by EPTI, that is Ethiopia Health um, Ethiopia Public Health Financial Services. You understand? Now, this is a nuclei of representative amoeba. Now, look at it. Can you see for histolytica, it is centrally placed. For coli, it is not central, it is eccentric. Can you see? It is eccentric. For this one too, it is central. For this one, it is nearly central. Can you see? And for this one, it is central as well. So this is eccentric, this is central. See? Let me use. This is eccentric. This is what? This is central. Understand? Now, we said, look at the next image here. This is an amoeba found in the stool of human. Understand? And it is like look at it. We have Entamoeba, Pispa, and histolytica. This is the trophozoite form. This is the cystic form. We have Entamoeba amani. We have Entamoeba coli. We have Entamoeba poleki. We have Entamoeba nana, and we have Entamoeba buski. These are what? These are images of these organisms. You understand? And let's talk about the morphological differences between each organism. The morphological differences between each organism. Now, if you notice very well, I have mentioned some morphological differences between them, and this is a table that shows it. This is a table that shows it. For I'm going to show the table now. Let me show the table no longer. Now this is Entamoeba gingivalis. This is an Entamoeba as well. These are images of amoeba. You understand? This is the chromatin body that I was talking about. You understand? Now I have an image. Actually, yeah, this is an image that shows what? Look at it. So this is a morphological difference between what? Between Entamoeba histolytica and other species. Now look at Entamoeba coli. The size of their trophocytes is about 20 to 30 micrometer, usually about 50 micrometer. You understand? And for histolytica and dispa, they have the magna form and the minuta form. The magna means the big form, and the minuta means the small form. Can you see? Can you see the measurement? Can you see the difference between measurements? And the cytoplasmic structure. The E. coli is coarser and granulated. The histolytica is finely granulated. The poleki finely granulated, and the gingivalis coarse and finely granulated. Understand? The position and the size of their endosome. One is eccentric, one is centric and large, one is central and small. The poleki is central and small, and the gingivalis is nearly central. So you can just try to screenshot this um particular image that shows the what? That shows the difference between the Entamoeba histolytica and what? And other species of amoeba. You understand? So this is what? This is this is the morphological differences. Can you see? Morphological differences. The size of cyst. The Entamoeba poleki and gingivalis has no cyst. It has no cyst. Are you getting that? And the number of nuclei. Now look at this. Very, very important. The number of nuclei. Usually above 5 and mostly 8 for this one. This one is 4. This one is only 1, and this one has no cyst. Now look at this. Look at this. This one has only 1 nucleus. And this is what? This is Entamoeba histolytica. It has only 1 nucleus. So look at this one. Has what? Has 4. Can you see? Has 4. This one has about 8. 1, 2, 3, 4, 5, 6, 7, 8. Yes, it has about 8. I see. One has 4, the other one has 8. So you can call this one Entamoeba dispar and this one to be Entamoeba coli. You understand? So that is it. That is it. So look at this one too. This is, I think this is Entamoeba. This is Entamoeba dispar or histolytica. Forgotten. But so these are what? These are the morphological differences between the two of them. Can you see this one? Entamoeba coliaci, Entamoeba histolytica as 4, Entamoeba coli as 4 as well. So this is what? This is the major differences between them. Now, in the next video, we will talk about the life cycle of Entamoeba histolytica, and that is what we will talk about in the next video. Thank you for listening and follow for more.